postdocs. The first one, University of Riesenberg in Germany, with Professor Hans Robert Kalbitzer, followed by a second one in Japan at Kinki University with Professor Kazuyuki Akasaka, and a third one in South Korea and Busan National University. And his research interests are in the areas of multidimensional NMR, variable high pressure NMR, solid phase peptide synthesis. And he has published a number of papers in solid state, solid supported or polymer supported peptide synthesis, protein NMR in this area. And today he will be discussing on variable high pressure NMR. With this short introduction, I invite Dr. Sunil Kumar. Dr. Sunil, please.
Yeah, thank you, Anil Kumar sir, for this introduction. So, good afternoon, all. So, we are starting with the last session of our 4 days NMR webinar. So, first of all, I would like to thank Arun sir and Anil Kumar sir for giving me an opportunity to give a talk in this webinar. And during the last 3 days, 3.5 days, we heard many interesting talks about the basics of NMR and different aspects of NMR, one-dimensional, two-dimensional, solid-state NMR, NMR instrumentation, and protein structure determination using NMR, multi-dimensional NMR, and small molecule structure determination. All aspects have been discussed during the last 3.5 days. Now, I am going to discuss about a different application of NMR spectroscopy, particularly in protein science. How we will see in detail. So, it's a variable high pressure NMR spectroscopy. So, before going into the details of a variable high pressure NMR, I would like to say a few words about our institution, Institute for Integrated Programs and Research in Basic Science, IIRBS, Mahatma University. And it was established in 2009. And we offer different courses there, in the 5-year integrated course, MSc course, master course, in chemistry, physics, biology, computer sciences, and environmental sciences, 4 seats each. Then doctoral degree in all these disciplines. Then we have an instrumentation facility, and instrumentation facility consists of a 400 MHz Bruker NMR spectrometer, HPLC, UV visible spectrometer, IR, atomic absorption, GCMS, Q-TOF LCMS, capillary electrophoresis, PCR, polarimeter, live laser, etc. So now we are coming to the topic high pressure science. So this is the general attitude towards pressure. We often want to run away from pressure. But pressure as a thermodynamic variable, it's very useful. And there are many branches in science related to pressure. High pressure NMR spectroscopy, high pressure micromolecular crystallography, high pressure fluorescence spectroscopy, high pressure small angle X-ray scattering, high pressure food sterilization, and different branches are there related to pressure as a thermodynamic variable. And all these topics are well described in a book edited by Professor Kazuyuki Akasaka, with whom I was working, High Pressure Bioscience, Basic Concepts, Applications, and Frontiers, published in 2015. And this is a picture took when he visited Kerala. I think it's from Munnar, something like that. So, and he mainly developed the technique, high pressure NMR spectroscopy. So coming to the high pressure NMR spectroscopy of proteins. So proteins exist in solution as a dynamic equilibrium of different conformation states with varying populations. So we have different experimental techniques for determining the structure of proteins. So I told you there are many conformation states, more than one conformation states, which are in equilibrium. Starting from their populations are varying. The most populated one will be the basic folded state. And then the other conformations, they are having very low population. And finally, we come to the fully unfolded state, which has even at normal state condition, even at under physiological condition, the fully unfolded state is present there in a very, very small population. So when we use X-ray crystallography for determining the structure of a protein, we crystallize the most populated conformation state, the basic folded one, and determine its structure. And we neglect the other low populated hidden conformations. When we do normal NMR spectroscopy, the conventional NMR spectroscopy methods, we determine a population weighted average structure of all the conformations which are in dynamic equilibrium. So this structure is very close to the structure of the most populated conformation. So here also we are neglecting these low populated hidden conformations. So variable pressure, variable high pressure NMR spectroscopy is very efficient for populating these hidden conformations, detecting them and characterizing them. So in variable high pressure NMR, we are not applying pressure to the NMR spectrometer. So we are applying pressure to the sample protein solution, and we observe what is happening to this protein in solution using NMR spectroscopy. So there is nothing special with NMR, high pressure NMR. NMR is the same, but we are applying pressure to the sample solution, protein or any other molecule. And then we observe, or then we monitor what is happening to these protein molecules or different conformations in this solution. So what is the effect of pressure on protein conformation states? So when we apply pressure to later we will see the experimental setup for applying pressure to the sample. So before that, what is the effect of pressure on conforming protein? So when we apply pressure to a protein in solution, mainly two things are happening. One is the general compression of each conformation. I said there are more than one conformations, and each conformation substate will undergo a general compression like this. And the second important thing which is happening is a conformational equilibrium shift will happen. Initially, some conformations are most populated, and there are very low populated hidden conformations. So when we apply pressure to this system, the conformational equilibrium will shift to the other conformational state. I will tell you how this happened. And then now we populated these hidden conformation states. Then we can use our NMR spectroscopy for characterizing these pressure-induced populated states in this protein solution. So the effect of pressure on the conformational states of proteins can be explained based on volume theory. So we know this Le Chatelet principle. So when we apply pressure to an equilibrium system, the equilibrium will shift towards the lower volume side. Here also the same thing happens. So I told you two things are happening. One is the general compression of the same conformation or same substrate. And this, a single substrate, a conformation substrate, It's an ensemble of conformations. They're having comparable energy and slightly different partial molar volumes. So when we apply pressure, the different conformations in the ensemble of a substrate, they undergo a shift. And this is giving, this is responsible for the general compression of a particular conformation in this equilibrium. The second thing what is happening is a conformational shift, equilibrium shift from one conformational state to another one. So in a protein solution, among different conformational states, starting from basic folded state to unfolded state, it's shown here, the potential energy level diagram. The most folded, the highly folded, basic folded state is having the highest partial molar volume. Then, when coming to the fully unfolded state, it is having the least partial molar volume. So when we apply pressure to such a system, we can shift the equilibrium from the fully folded state. So under normal conditions, the fully folded state is the most populated state, say 90%. The other are coming in a 10% population. So when we apply pressure to the system, the equilibrium will shift towards the low populated conformational states. Finally, after a very high pressure, it depends, the pressure level depends upon the nature of this protein. All the proteins will come into a fully unfolded state, which is not functional state. So this is the explanation for the effect of pressure on protein conformation state. It's based on the difference in partial molar volumes between different conformations. If there are no difference in partial molar volume between different conformational states, the protein will be insensitive to pressure. We cannot shift the equilibrium. Just like NMR active nucleus, if the nucleus is not having any spin, it's NMR inactive. Like that. If there is no difference in partial molar volume between different conformational states, we cannot shift the equilibrium. We cannot populate low populated one. So this is a variable high pressure NMR setup or system at the University of Regensburg, Germany. So we used an 800 megahertz spectrometer with a cryoprobe. And this is a normal spectrometer, as I told you. But here there is a special setup for high pressure, for applying pressure to the sample. So we need a pressure generator. The pressure generator can be manually operated and it can be automatic. So for this, in the case of this manual pressure generator, we have to change the pressure after. So we have to record NMR spectra at different pressures. We have to vary the pressure. So we have to change the pressure, increase or decrease the pressure manually. But in the case of this automatic pressure generator, we can control it using a computer. And we can give all the pressure levels which we need, and it will automatically change, increasing or decreasing, depending upon the values which we gave to the program, computer program. And there is a pressure line between this pressure generator and in variable high pressure NMR spectroscopy, we use a special NMR sample tube. Here it is ceramic NMR tube, which can withstand very high pressure up to our laboratory. We had in this laboratory, we had a ceramic cell which can withstand up to 3,500 bar or 350 megapascal. So a picture of this ceramic cell is shown here, and this is connected to this adapter. And there is a pressure line connecting between this pressure generator and this sample tube. And this is usually water. And we use a membrane for separation, for separating between sample and this pressure mediator. So we can apply pressure to the sample. So we can vary the pressure, increase or decrease, and at each pressure we can record different types of NMR, and we analyze what is happening to the spectra when we change the pressure. That is what we do here. So in the case of an unlabeled protein, this is a small spectrum from a small protein, CMR to subtype. We will see as an example later, details. So in the case of an unlabeled protein, unlabeled means we don't have any N15 or C13 labeling. You know what it means. So Sulakshana and others explained in the case of proteins what is spin labeling and what it is doing. And we can record one-dimensional proton NMR spectra as a function of pressure. And we can analyze the peak position. Peak position means proton chemical shift, and peak area also. We can analyze how they are changing as a function of pressure. So suppose we have an N15 labeled protein. So we can do heteronuclear HSQC, proton N15 HSQC, and we can record different HSQCs as a function of pressure. We vary, for example, starting with 30 bar, then we record an HSQC, increasing to 500, 1,000, 150, like that. So now we all know what is an HSQC, and each cross peak represents an amino acid. That is coming from, this is a correlation between proton and N15, nitrogen, amide correlation, proton N15 correlation, each cross peak. So each cross represents an amino acid in the protein. This is from ubiquitin protein. And see, we can clearly see that the cross peaks are changing with the pressure. They are shifting their position, and some of them are changing their peak volumes, like that. That means this protein is sensitive to pressure. Then we can analyze the peak positions. That means we can do proton, we can get proton and N15 chemical shifts at each pressure, and we can get peak volumes at each pressure as a function of pressure. That is what we do usually. Then, so what are the different types of pressure-induced spectral changes we usually observe? So there are different types of spectral changes, pressure-induced spectral changes, which we usually observe. What are they? The first one, change in peak position, that means chemical shift, without any area or volume change. Usually two types of such changes we see. One is a linear change, which indicates that fluctuations within the same conformational substate, the same folded state is fluctuating. That is indicated by this linear chemical shift change. And the second one, the green one, this is a nonlinear change, which indicates the occurrence of two or more conformations in the equilibrium, which are in fast exchange, and their populations are changed by pressure. And here it is a two-state model. That means only two conformations. This is a sigmoidal curve. State one conformation is changing to the other one. So in general, the nonlinear chemical shift change indicates that the equilibrium is shifting from one state to the another. That means there are more than one conformational states which are in fast exchange, and pressure is changing their equilibrium. It is shifting from one state to the other one. So the second type of spectral change which we observe, there is no change in peak position. That means no chemical shift is changing. Only the area or volume is decreasing or increasing with pressure. Normally it is decreasing. That means there are more than one conformations in this equilibrium, and they are in slow exchange. A population change, the peak volume decrease or a peak volume difference or change in peak volume represents a population change. And it indicates occurrence of two or more conformations in the equilibrium which are in slow exchange, and their populations are changed by pressure. Here is a plot of normalized peak volume as a function of pressure. We get a sigmoidal curve, that means two states are in equilibrium, they are in slow exchange. So the second, the volume of the original peak is decreasing. In some cases, we observe a second peak which is appearing at a particular pressure point, and the volume of that the second peak, the new peak, is increasing. That means population of the second conformation is increasing at the expense of the first one. In some cases, we don't observe the second or the new peak because of overlapping with the other peaks. In the spectrum, there will be many number of cross peaks. It depends on the number of amino acids. For line broadening, if the second peak is has a very large line broadening, then it is usually not visible. It will mix up with the noise peaks. So we are going to the next third type of spectral change which we usually observe. In this case, both chemical shift and peak area or volume are changing. Say linear and non-linear chemical shift change. But in both of the cases, volume is decreasing as a function of pressure. It indicates the occurrence of more than two conformations. The chemical shift indicates that what it does indicates there are some conformations which are in equilibrium, some of which are in fast exchange. The chemical shift change as a function of pressure indicates that the population of the conformations which are in fast exchange, it's changing with pressure. And the peak volume change indicates that the conformations which are in slow exchange, their populations are changing by pressure. So there are more than two or more than one conformations or three conformations, for example. Some are in fast exchange and some are in low or slow exchange. And the chemical shift change indicates the equilibrium shift between conformation states which are in fast exchange. And this peak volume change indicates between conformations which are in a slow exchange. So usually we observe the peak width. The broadening will happen based on the exchange rate. So if the exchange rate is fast, then we get a sharp peak. When the exchange rate is in the microsecond scale, then we get a single peak from all the conformations. And when it becomes reaches up to millisecond scale, then the broadening will happen. If the exchange rate is even increasing to second or more than that, then we will get two peaks. The separation will happen. So then now we come to the details, further details. So I told you this linear type of chemical shift change as a function of pressure indicates fluctuations within the same conformation state. And the magnitude of such fluctuation at each point can be estimated from delta delta. So how much chemical shift difference it is changing. So if there is a higher value of delta delta, higher the flexibility, that is fluctuations within the same conformation state. So transition from one conformation state to another, which are in fast exchange. So this information we get from the nonlinear chemical shift change. So this is a two-state model chemical shift change. State 1 is going to state 2. which are in fast exchange. We cannot separately see their peaks. But we can shift the equilibrium from state 1 to state 2. Here in this case, at around 200 megapascal, state 2 is the populated one. And we can record spectra at this pressure. And we can characterize the state 2. At a normal pressure, at ambient pressure, the state 1 is most populated. So we can characterize it. And at a higher pressure, we can populate the other one and characterize this conformation state. So we can estimate the free energy difference and partial molar volume differences between these different conformation states by applying some thermodynamic equations we will see later, which are there. So from my experience and from other high pressure people, we can see three-state model. State 1 is going to 2 and it is going to 3, like that. And by fitting corresponding thermodynamic equations, we can get the free energy difference and other thermodynamic parameters between of different protein conformation states. So for estimation of linearity and non-linearity of chemical shift changes, so we can get linear and non-linear pressure coefficients by fitting this equation. Delta I is equal to AI plus BIP plus CAP square to the plot of chemical shift change as a function of pressure. This can be a proton chemical shift or a nitrogen chemical shift. Then here delta I P is the pressure, delta I is the chemical shift at a particular pressure, and AI is the chemical shift at 1 bar, that means atmospheric pressure. And BI is the linear or first order coefficient, and CI is the second order coefficient. So we can estimate the linearity and non-linearity of pressure induced chemical shift change from BI and CA. So conformational fluctuations in tau a slow time scale. Changes in cross peak volumes indicate conformational fluctuations in slow time scale. This we already discussed. So we have a peak, and with varying pressure, its volume is increasing, and a new peak, in some cases, a new peak will come, will appear, and we call it split peak or new original peak and split peak, and its volume is increasing with pressure, and some of the volumes, the total sum of these volumes at each pressure will remain constant. And the new split peak may not be visible due to overlapping or throttling. So now we are coming to the thermodynamics of variable high pressure NMR. So the equilibrium constant K between different conformations, here it is N and I, may change with the pressure according to the, so when the population changes, the equilibrium constant will also change. K is equal to concentration of I divided by concentration of N, which is again equal to e raised to minus delta G by RT. And the free energy difference, delta G equal to Gi minus Gn equal to delta G0 plus P delta V0. Here delta G is the free energy difference at pressure P. Delta G0 is the free energy difference at ambient pressure, normal atmospheric pressure. And delta V0 is the difference in partial molar volume between the two conformations, N and I, at ambient pressure. And R is the gas constant and T is temperature. Usually the change in delta G as a function of pressure is linear. So we can plot delta G as a function of pressure, and we get a linear plot here. See, it obeys this equation, delta G is equal to delta G0 plus P delta V. The slope of this linear curve is delta V, the difference in partial molar volume, and the y-intercept is the delta G0, the free energy difference at ambient pressure. See, at some pressure, the delta G0 or delta G is becoming negative. What does it mean? the population inversion is happening. The population of the second conformation is higher, becomes higher than that of the first conformation. For example, initially N was the most populated one, I was the low populated one. At some pressure point, the reverse is happening. That means I becomes, the conformation I becomes the more populated one, and N becomes less populated one. So now we have different types of chemical shift changes as a function of pressure. 2-state, 3-state, 4-state. Usually we don't see 4-state, 3-state, different types. All these represent for conformational equilibrium shift between conformational states which are in fast exchange. And we have different thermodynamic equations for fitting to these chemical shift changes as a function of pressure. And we can get delta G0, delta V0 between different conformations, and delta G0, delta G12, delta G13, like that between different conformations. So now we come to the slow conformational state, pressure-induced peak volume changes. So there are two cases. Only one peak is visible. That means its volume change can be monitored. Say the volume is changing like this, and it is a 2-state case. We can fit this thermodynamic equation given here to this volume, a plot of volume as a function of pressure. And we get delta G0 and delta V0. In the second case, both of the peaks are visible, the original peak and the split peak. In this case, the equilibrium constant at each pressure can be estimated by taking the ratio between the peak volumes of the original and the split peak. So K is equal to VNP by VOP. Here, VNP is the volume of the new peak, and VOP is the volume of the original peak. And again, delta G is equal to minus RT dot K. So we can plot delta G as a function of pressure like that. These are the different concepts in a variable pressure, high pressure spectroscopy. Now we are coming to some case studies. So in shortly, I will discuss about 3 proteins, and we did variable high pressure NMR spectroscopy studies with these 3 proteins. We did with many, and I would like to explain only these 3 here. And the first one is CMIP R2 subdomain, and this is an unlabeled protein. So we used a one-dimensional proton NMR spectra as a function of pressure. And the other one is a RAS protein, and this is a GTPS protein. We will see it one by one. And the third one is E. coli DHFR protein, dihydrofolate reductase, and it reduces dihydrofolate into tetrahydrofolate, and which is used for synthesis of purines and pyrimidines, which are again used for DNA synthesis. So, coming to our first example, this is the case of CMIP R2 subdomain, and the title is given like this: extensively hydrated but folded, a novel state of globular proteins. And all these results are published in Biophysical Journal in 2012. Okay, so these results give 2 important messages. One, we should not come to a conclusion with only one type of experimental method. This is a message to the young generation of researchers. And the second message is discrepancies between different types of experimental methods applied on the same system, for example, may lead to exciting results. Sometimes we do different types of experiment, or we apply different types of experimental methods to the same sample, and we see discrepancies, and we leave it there. But if we can connect them, correlate them in the meaningful way, we can get exciting results. So we will see such an example here. So protein unfolding is a wide protein folding and unfolding. So we use different methods for experimental methods for monitoring the uh, what the unfolding of a protein. One method is tryptophan fluorescence. So tryptophan side chain is highly hydrophobic. So in a folded protein, these tryptophan side chains, they want to occupy the core region of this because it is hydrophobic. They want to be away from water. So they usually occupy the interior of the protein, the core region of the protein, a folded protein. So if you do so the emission maximum, the fluorescence emission maximum, lambda maximum, it comes around 335 nanometer under this hydrophobic environment. Suppose the protein is undergoing unfolding by some kind of perturbation, the lambda maximum will shift to a higher value, 355, around 355 nanometer. That means the protein is in fully unfolded state and all the tryptophan side chains are hydrated, fully hydrated. So this is one very useful method for monitoring the unfolding of a protein. The second method is also again very efficient, is using NMR spectroscopy. So we have peaks, NMR peaks, corresponding to the folded state. So by applying some perturbation, we can shift the equilibrium from folded to unfolded state, N to U. So when it is happening, the peak volume or peak area corresponding to the folded structure will decrease. And finally, it will become zero. That means there will be no native state or folded state in the system. All the proteins are in an unfolded state. So NMR also is a very efficient method for monitoring the protein unfolding. So in this case, in this study, what we did, we have applied variable pressure tryptophan because here the perturbation we did, we applied is pressure and we changed the pressure. So we applied variable pressure tryptophan fluorescence and proton NMR to CMIP R2 subdomain at low temperature, 5 degrees Celsius, for knowing the conformational changes along the pressure axis, the thermodynamic axis. Coming to CMIP, it's a transcription factor involved in proliferation and differentiation of hematopoietic cells. It has three functional domains: DNA binding, transcriptional activation, negative regulation. And the DNA binding domain itself has three subdomains: R1, R2, and R3. The three-dimensional structure of R2, our subject of interest is R2, and it has 52 amino acids. And when we look into the three-dimensional structure and analyze it carefully, we can say that there is a large internal cavity in the core region of this R2 subdomain. And it's more dynamic nature, and it is less stable compared to R1 and R3. So we can say that this internal cavity is responsible for these differences, particular nature of R2. So in this study, what we do, there is a residue, valine 103. And its side chain is directly oriented into this cavity. And when we mutated this valine with the leucine, with a long side chain, the cavity size was reduced. So our aim is to have two proteins. One is with a large internal cavity, and the other one is with a small internal cavity or without any internal cavity. In this case, that's a very small one. And we can see that all the tryptophan side chains, all are colored red, are occupying the hydrophobic core of this R2 structure. So coming to the experimental part. So there is a cysteine residue in this protein. So this cysteine, it is always making problems by oxidation, dihybridization, and all these kind of things. So we avoided this cysteine by substituting with isoleucine. But this substitution did not change any, did not affect the cavity size. So the C130I mutant of wild type CMYC2 is considered as the pseudo wild type WT star. It has large internal cavity. And the second one is the cavity filled V103L mutant of WT star. Then what we did variable pressure tryptophan fluorescence from 3 megapascal to 600 megapascal at pH 7 at temperature 5 degrees Celsius. Then with both of these proteins on this, then what we did, variable pressure one-dimensional proton NMR from 3 megapascal to 300 megapascal at pH 7 at temperature 5 degrees Celsius. Same conditions, only pressure is very. Okay, this is the online. I did all these experiments at Kingi University, Japan, in association with Professor Kazuki Akasaka. And this is the variable pressure fluorescence spectrometer. It's a setup there. And this is there, and there is a 600 megahertz spectrometer. And this is a pressure line, and the ceramic cell, everything is here. NMR, these online means during the experiment we are applying pressure to. Previously at the beginning, what scientists were doing, they applied pressure in a closed vessel and then took it and closed very tightly and took it to the spectrometer. There was no experimental setup for online pressure at the same time. But now the technique is much developed, and we can apply pressure at the same time of experiment. And we can vary also. Coming to the results. So from tryptophan fluorescence, we can see that at around 300 megapascal, the lambda maximum emission has reached around 355 nanometer. That means all the tryptophan side chains are fully hydrated. So if we stop at this point, we conclude that it's fully in an unfolded state. The protein is unfolded, and all the tryptophan side chains in the interior, they are fully exposed to water. That is why this shift happened. Then we did a variable high pressure NMR on the same sample. So this is, we are discussing about this wild type with the cavity. See, we can see that even at 300 megapascal, we can see peaks, NMR peaks from folded state. That means the protein is in folded state at 300 megapascal. It's not in unfolded state. So when we combine these two results, we can say that there exists a conformation which is fully hydrated or extensively hydrated. And we are shifting the equilibrium from the non-hydrated to the hydrated conformation state. So when the interior of the protein is in the folded state, but the interior is fully hydrated, water is entering into this, or the fully interiorly hydrated conformation state, the population is increasing. So hydration of tryptophan side chains without unfolding is possible only if water penetrates into the interior of the protein, or if only if, so even at ambient pressure, there is a small population of this extensively hydrated conformation in which the interior of this protein is fully hydrated. But the most populated one is the other one. So by applying pressure, we are populating, or we are shifting the equilibrium to the extensively hydrated folded state, folded conformation state. So we are populating that and characterizing that. So an extensively hydrated folded state, N prime, occurs in R2, which is in equilibrium with the basic folded state N. And we can shift the equilibrium by varying the pressure. So the presence of the second state N prime is supported by the nonlinear nature of pressure-induced chemical shift change. We observed many nonlinear chemical shift changes, Zig model and other kind of things. This is supporting our observation. And again, so when we come to the cavity-filled case mutant, see even at 600 megapascal, the tryptophan fluorescence is staying at a lower value. That means the tryptophans are not fully hydrated. That means no such fully hydrated conformational state is existing in this case of cavity-filled mutant. So this internal cavity is intimately related to the existence of extensively hydrated folded state in CMIP R2 subdomain. So by fitting our thermodynamic equations to the changes in lambda maximum as a functional pressure, we estimated delta G0 and delta V0 between different conformation states, coming to this conclusion. So the apparent discrepancy between the variable pressure tryptophan fluorescence and variable pressure proton NMR results shows the presence of an extensively hydrated folded state in a globular protein seen in R2 subdomain. The basic folded N state of CMIP R2 is in equilibrium with an extensively hydrated folded state N prime. The internal cavity is responsible for the existence of this N prime state in R2. The N prime state is more heterogeneous than the N state, as evidenced by the line broadening in proton NMR signals with pressure. Interpretation of the conformational stage of a protein based on tryptophan fluorescence alone may not be complete, but a combination of NMR spectroscopy is more informative. So when we come to the biological implication of these results, so it has been reported that by Professor Ogata and all from University Hiroshima University, and it has been reported that the tryptophan 95 side chain upon DNA binding, this is a transcription factor, so it is binding to the DNA. Upon DNA binding, slides into the internal cavity present in R2 subdomain, which requires high conformational flexibility of R2. So we speculate that the more fluctuating, extensively hydrated conformer, N prime, is more likely to be involved in DNA binding. That means we identify the real functional state of CMIP R2 using NMR spectroscopy, a variable high pressure NMR spectroscopy. So now we come to the second example, in the case of RAS protein. So using high pressure NMR, and all these results are published in Angewandte Chemie in 2013. And we did a variable high pressure NMR spectroscopy on RAS protein. The GTP bound, we will see the details soon. And we identified different multiple conformation states in it. So RAS is a small GTPase. It can hydrolyze GTP. And it is involved in transmitting signals within cells, intracellular signal transduction. So in its functional cycle, the RAS cycle, RAS is shuttling between GTP bound and GDP bound. GTP bound form is the active form, the other one is inactive form, GDP bound. And during this RAS cycle, it has to interact with many other partners, many other proteins are there in its cycle. Exchange factors are there, GTPase activating proteins are there, then RAS effectors are there for transmitting the signals, like that. So when we analyze this RAS cycle, so there should be at least 8 drug-relevant functions, functional states of the protein that can be used for that we will discuss later. we say that at least when we consider both GDP bound RAS and GTP bound RAS, we can say that there will be 8 different conformational states. So if we consider only the active GTP bound form, we can say that it can have 4 different conformational states. Each state can interact with a particular interacting partner. Not the same conformation is interacting with all the partners. So when it interacts with a particular interaction partner, it adopts a particular conformation, and a different conformation when it interacts with another partner. That is true. So in the case of GTP bound form, we expect at least 4 conformation states. So what we did, we have an N50 labeled protein. So instead of GTP, we used a GTP. There is a possibility for slow hydrolyzation. So during the experiment, we need long experimental time. It may undergo hydrolysis and may disappear from the system, GTP. So what we used a stable analog of GTP, GPP and HP. And we took a complex between RAS protein and this magnesium is the cofactor, which is necessary for its binding. And we used an 800, which I showed you, spectrometer with the cryoprobe. And we recorded HSQC proton N15 HSQC spectra as a function of pressure. So an overlay of all these spectra are shown here. Then we analyzed the spectra as I explained before. We determined the linear and nonlinear coefficients of different both proton and N15 chemicals. So by fitting this equation, here it is B1 and B2, nonlinear and different notations are used in different groups. So we plotted the B1 and B2 values with very significant values, very high values on the structure. You can see different colors on it. Then we followed the volume changes, and some of the residues are undergoing much volume change as a function of pressure. That means some conformations are in a slow exchange. And we plotted or color coded residues which are showing considerable reduction in a peak volume. So we have a non-linear chemical shift changes, like 2-state and 3-state also there. That means there is at least 3 states which are in fast exchange. And there is a volume change which we already see. That means there are conformational states which are in slow exchange. And when we analyze the volume change or plotted as a function of pressure, we observed that 2-state modeling, the changes are observed. And we fitted these different thermodynamic equations for obtaining delta G0 and delta V0 between different states. And we observed that we observed 3 conformational transitions between the C2. C2 is the most populated one, which is the basic folded state. And it is undergoing transition to C1, and C3, and C4. Of course, this C2 to C1 and C2 to C3 are fast. They are in fast exchange, and C2 to C4 is in slow exchange. That is clear from the volume change. And the thermodynamic parameters or free energy difference between, for example, C2 and C1, and C2 and C3, C2 and C3, they are estimated by fitting thermodynamic equations. And we got these values. And now we know which are sensing. We know which residues are sensing C2 to C1 transition. And they are color coded here on the three-dimensional structure. And C2 to C3, and then C2 to so4. Some residues are sensing multiple conformation transitions. we can estimate the population of different conformational states at different pressure as a function of pressure. At normal pressure, C2 is the most populated. C1 is there, and with the pressure, population of C2 is decreasing, and C1, another conform, we'll see here, and C1 initially increasing and decreasing. C3 is increasing continuously. C4 is very slowly increasing. And here in this figure, it is given which is 3T. 3T is interacting with this GTPase activating protein, another interacting partner, this state. And the 1T, 1T is interacting with this exchange factor, and 2T is interacting with the effector. And the state 1,0, it is a nucleotide free state. So it's a state existing between. So GTP is hydrolyzed to GDP, right? So there is a transition between GTP bound and GDP bound forms. So this 1,0 state exists between them. It's a nucleotide free. So we identified all the 4 states. What is significance of this? So we know which residues are involved in a particular conformational transition or in a particular state. So since cycling, this study was done by Ina Rosnevic from the same group of Kalbitzer, published in Agave and Academy. They found that zinc cycling can bind to RAS. It binds to 1T state or C1 state, which interacts with the GEF. That means, so do you know how? So this RAS protein is an oncogenic protein. So in its normal state, it's enough for cell signaling. So some mutations, and it is responsible for cell divisions and proliferation and all these kind of things. So when it becomes permanently active by some mutation, this protein at some point. So almost 30% of cancer occurring in human is from this mutation happening on RAS protein. So the permanent activation of this protein is dangerous, leading to a cancerous state. So this state 1 or 1T state is interacting with the exchange factor. That means, and this factor is helping the exchange from GDP to GTP. So the GTP bound form is the active form. GDP bound form is the inactive form. So when this zinc cycling binds to, if it binds to this state 1, it stabilizes it. That means this exchange is not happening. So it can prevent it from GEF mediated activation. That means can control the cancer state. Conclusions. When a protein interacts with the different proteins in a function cycle, different structural states are required for a specific recognition of the partners. In general, these structural differences are not restricted to the interaction site itself, as our high pressure data nicely shows for activated RAS. These structural differences within or outside the protein-protein interaction surfaces create intrinsic allosteric binding sites that can potentially be recognized selectively by small clickings. In summary, this non-classical type of allosteric interferences provides a generally applicable strategy for the modulation of protein-protein interactions by small compounds, independent of the existence of classical allosteric regulatory sites. So, in conclusion, we can use this strategy for developing new drugs. So, the drug molecules can target each state and modulates its function very effectively. So now we are coming to the third protein. It's a dihydrofolate reductase. It's an enzyme. So it reduces folate into dihydrofolate into tetrahydrofolate. As I mentioned earlier, this tetrahydrofolate is used for the synthesis of thymine, pyrimidines, purines, and pyrimidines, which are again used for DNA synthesis. So the cofactor of this catalytic cycle is NADPH. Okay, this is a three-dimensional structure of dihydrofolate reductase from E. coli. We also have E. coli. This dihydrofolate, all species, 233 species from bacteria to human, all species are having this E. coli, this dihydrofolate reductase enzyme. So the folate is bound to this enzyme shown here, and there is a NADPH, the cofactor binding site is there. So near this cofactor binding, NADPH binding site, there is a loop, we call it as a M20 loop. So Professor Kitahara and his group, and he was also associated with Professor Akasaka, they found that. So what I want to tell you, see, in the absence of NADPH, this cofactor, this M20 loop can orient in two different ways. Either it can occupy this binding site, that state we call occluded state. And in the other case, this loop is, M20 loop is away from this NADPH binding site. That means the open conformation. And this open conformation is needed for the binding of NADPH. So this is the function state. And this Kitahara-san and his group, he found that even at an atmospheric pressure, a small population, approximately 10% population of this open conformation is existing, which is in slow exchange with an occluded conformation, which is having 90% population. In the case of wild type DHFR. In our study, what we did a single mutation at a position glycine 67. What is the peculiarity of this mutation site? So this the active residue is aspartate 27, D27. And this glycine 67 is occupying on a loop which is very far away from this active site residue. We cannot tell anything. We cannot say that there is some correlation with this. It is very far away. But in order to understand the effect of mutation on this remote site, we did a glycine 67 valine mutation. Glycine was replaced with valine. Then we did variable high pressure NMR. Our aim is to understand how this open occluded conformational equilibrium is shifting by this mutation. Is there any effect of this mutation on this equilibrium? So we did variable high pressure NMR. We recorded many HSQCs, proton N15 HSQCs as a function of pressure for both wild type and glycine 67 valine mutants. See, we can see that some of the residues are splitting as a function of pressure. For example, valine 13, some residues, so the valine 13 is enlarged here. And another one is glycine 95, tryptophan 22. Then one more residue also there, glycine 51, like that. So that means at ambient pressure, one conformation was dominant. We observed the peak from that. When we increased the pressure, the equilibrium shifted towards the other side. The population of the other conformation increased. That is why a new peak originated or became observable. And we can see that the volume of the second peak is increasing with pressure. We will see in the next slide. And when we see the location of these residues, which are sensing which are showing peak splitting, it's around the NADPH binding site. See the red colored regions, the red colored bolts, they are showing this location of these residues which are showing peak splitting, which are uh, uh, very clear from this spectra. So these residues are locating or they are occupying the around the NADPH binding site. That means, and this splitting indicates the open occluded movement or open occluded equilibrium shift in a equal DHFR. Then now we want to know how this lysine 67 valine mutation affected the population of this open and occluded conformation. So Kirahara-san reported that at ambient pressure, the population of open conformation was, which is the active form, was only 10%. The glycine 67 valine mutant, the population was increased to approximately 18%. That means almost double. So there is a considerable effect on the conformational equilibrium of a protein, which is occupying a site which is very far from the active site. So we did many more experiments. We analyzed the conformational fluctuations in the whole protein, and I don't want to discuss this. And all the results will be published soon. The manuscript is under preparation now. And so this is an example for a slow conformational exchange between open and controlled conformation. So coming to the acknowledgement, I should thank my previous research supervisors, Professor Kazuyuki Akasaka and his group members, Dr. Akihiro Maino and others. Then from University of Regensburg, Professor Kalbitzer and other friends in the same group. And coming to Kyoto Prefecture, this same was supplied and by this Professor Masayuki Oda and his group. So, and I should thank all the members of IIRBS, Mohammed University, my students, other faculty members, and other members of IIRBS. So thank you all.
Thank you, Dr. Sunil, for a very nice presentation. It's, of course, the topic is quite relatively new and difficult to understand, but you have done it in a very simple manner. Now this is open for questions. The participants can put your questions in the chat box.
I can.
Okay, you can.
One moment, chat box. Yeah. Yeah. There are. Sir, can you use? Oh, sorry, I could not use this laser point. And sir, as we increase pressure, the number of molecules in the unfolded state increases, and we are getting our results for the unfolded state. How will we arrive at the folded structure, which is supposed to be the actual structure? So that's a very good question. So when we start with a new protein, what we do first, we optimize the pressure level. So there are peaks from the folded state and also from the unfolded state. And we stop the pressure at a level when the peak from the unfolded peak starts appearing. That means below that pressure level, the protein is in the folded state. Did you understand what I mean? So we are increasing the pressure gradually, step by step. So we have NMR cross peaks from the folded state of the structure. So at some point, the peaks from the unfolded state will start appearing. For example, in the case of one-dimensional proton, the peak around 0.9 ppm, they are coming from the medial side chains of leucines and lysines and all from the unfolded state. So we can clearly see that this peak will increase its area or height. So that means the protein is undergoing unfolding at that stage. So when we want to study a protein in its folded state, we have to stop the pressure at that level before starting unfolding. Okay, hope this is clear. Eh? Hello?
Maybe we can go to the next question.
Yeah. So next question is, will high-pressure NMR experiments of proteins be useful to study the mutational changes that occur in viruses, as we heard the case of the mutation coronaviruses? So in virus also there are proteins. So we can do high-pressure NMR in virus proteins also. But only condition is that protein should be sensitive to pressure. So I told you the equilibrium shift and all these kind of things will happen only if different conformations are having a partial molar volume difference. So if this protein, any kind of protein, virus protein or any kind of protein, can be studied using variable pressure NMR if it is sensitive to pressure. Okay, may I go to the next question. Will the application of pressure lead to the denaturation of protein? Yeah, of course. So I would like to explain one interesting thing. So I was also thinking like this. The unfolded state occupies in a conformational equilibrium or in a protein solution. I believed that the unfolded state occupies at extreme conditions. For example, at a very high pressure or a very high temperature or any other denaturing. But in reality, fully unfolded state is occupying even at physiological condition, but the population is very, very small. The peculiarity of variable high pressure NMR is it's not creating any new conformation. It's shifting only the equilibrium. So if there is no unfolded state in this equilibrium, even at normal pressure, we cannot shift that equilibrium. But we are able to shift the equilibrium to this unfolded state only because a very small population of unfolded state is occupying even at a physiological condition. So at some point, the population of this unfolded state will be very high. That is happening there. So how do you select which particular site to mutate and why what residue? That what residue? So in this case, there are many questions when we have a protein. Many questions will come to our mind. The first one will be about the structure. First, we will study the structure using NMR, for example. Then we will determine which are the residues normally involved in this function. For example, if it is an enzyme, it's a catalyst, biological catalyst. So there are some residues involved in its catalytic activity. So usually what we do some mutation around the catalytic site or the active site. and we check whether this catalytic activity is changing, increasing or decreasing or something like that. That is one way of selecting the mutation site. So in this case, in the case of dihydrofolate reductase, what we did, we selected a residue which is very far from the active site. Usually we think that this residue has no effect on its structure or function, something like that. So in a biomolecular NMR assignment, we published another paper. So we compared the structure of the overall folding, and it's not much affecting. But this open the overall structure is not, but the open uploaded equilibrium is shifting by this mutation. So the mutation site is selecting based on the purpose or which question we have to answer. This is the basis of selection. And in this case, we selected glycine substituted with valine. So valine is another hydrophobic residue with a longer side chain. There is no other reason for that. Is there any more questions?
I think there are no other questions. If there are any questions, the participants can put it in the chat, please.
I think there's no more question.
Okay, then I think no, I should once again thank Dr. Sunil Kumar for the wonderful lecture.
Thank you.
And also,
Thank you.
I take this opportunity to thank all the other speakers in the previous days and today. Thank you very much. And I think Dr. Arunan will have to say something about feedback and Dr. Arunan.
Thank you, Dr. Sunil. Actually, when I was planning for our webinar on NMR, I contacted Dr. Anil Kumar, and he immediately suggested Dr. Sunil Kumar would be the ideal person because he's the right man who is doing NMR. And suddenly everything settled down, and Sunil selected all these speakers. And from these 4 days of class, you might have noticed that all are very excellent speakers. And unlike any other teachers, see, most of the students who are working in college or studying in college, they know that whatever you learn from teachers is from the textbook. But these are the pioneers in NMR. So whatever you got is real knowledge, first-hand information from them. So I thank you, Dr. Sunil Kumar, for selecting the best speakers and best scientists for the topic. And of course, Dr. Anil Kumar also. He is my best friend. Whenever I plan to something for organic chemistry or chemistry programs, the one who I always approach him first, and then he immediately plan everything. Thank you, Dr. Anil. And so it was a wonderful webinar. I noted that almost above 500 participants people actually registered for, but eventually it turned out only 100, nearly 120 people. Those are the genuine candidates, I know that. And it's always like that. Only some people purely out of curiosity they register and they won't finally turn out. And I know that even these 100 people are benefiting out of these 4 days of program. We are really happy. Thank you so much. Thank you for attending all these 4 days. And we will give you the certificate for participation in 2 days. And that's all I wanted to say. I think we could conclude, right? Okay.
Yes.
Yeah.
Yes.
Thank you.
Okay. Thank you so much.
Sunil,
I can give you the feedback link also for the participants, right? So that we can take the materials.
Sunil, you want to say something more?
No. Thanks to nothing more.
Maybe we can have one more webinar maybe in 6 months' time.
Yeah, sure.
Yeah. Sure.
So in 2 or 3 weeks' time, we will have another webinar on instrumentation, especially instruments that are used in material science, that covers IR, Raman spectrophotometer, and scanning electron microscopy and all. So that will be useful for chemistry as well as physics students. So probably some of you can join there. I mean, the participants can join there. Anil or Sunil, if you want to share something, you can have.
Yeah, thank you all. We can talk. Yeah.
Thank you very much.
Thank you all the participants. Okay, bye then.
Okay. Bye-bye.
Bye. Bye.